The first session, which had the task to bring together some factual information on the current knowledge, but as I said earlier, also on the gaps in our knowledge. And I think that all our speakers have done a marvelous job in not only doing the description of their relevant subjects, but also highlighting what are the questions to be discussed over this today's and the questions for research to be done in the future. Now, we're coming to the question and answer part of this session. And I would kindly ask those of you who have questions that they introduce themselves with a name and affiliation or with the relation to the subject, and that they also say to whom the question is directed. Would somebody be so brave to start? I saw the first one just straight near the window. Do you have a microphone at the very end of the room in the middle of the windows?
Okay. Hi, my name is Dr. Gail Comer. I'm a gastroenterologist working in Pfizer R&D with a number of monoclonal antibodies. And it was of great interest that Biogen is developing an antibody to JC virus for commercial testing. And I was wondering how this compares Eugene Major to the one being developed at the NIH, and what are the things we should be looking for in such an assay in terms of being able to understand who was previously exposed to the JC virus and might be at greater risk of developing PML with immunomodulation therapy.
I think that question was directed to me, I guess.
Yes, it was.
So I think most of the current laboratories that have antibody assays to detect antibodies to the presence of JC are ELISA-based assays. The antigen that is used generally is what are called virus-like particles. In that second or third slide that I had, and I showed you actually the virion structure. Actually, that is a clone of those sequences that make the VP1, and that makes those proteins self-assemble into viral-like particles, VLPs, similar to what papillomavirus researchers did to make the vaccine. That's the antigen that coats the plates. I think, and I think the Biogen representatives here could talk about their assay. But that generally has been the antigen that has been used in a standard ELISA format for the detection of the presence of antibodies to JC. There certainly is increased interest in the technology, the methodology that one laboratory uses compared to another laboratory, how the data are reported out, for example. We like to report out data when we run our ELISA assay against VLPs using positive and negative controls. We like to report that data out in terms of titer. And I had mentioned that in my talk. In other words, we make four-fold serial dilutions, and we run these out on standard 96 well, plates. And so we will say, for example, in our assay, that we consider a sample to be serologically positive at a titer of 2560 or greater. Below that, then we would consider that a sample that would be negative. And we benchmark that against other ways that we look at the ability of the presence of antibody to inhibit hemagglutination, which is a characteristic of this virus or not. I think it's probably important, and I could say this taking advantage of your question, that we take a look at different laboratories who use these kinds of assays, who develop the assay, and at a time I think it's come so that we can all share a common understanding of exactly what that information means. And for example, should we all use the same antigen? I think that's a very important question. Should we all use the same technology and methodology format? And how do we arrive at the definition of what we consider serologically positive or serologically negative? But I think generally, just to be specific to answer your question, I think almost all assays are ELISA-based, using the antigen that I described as viral-like particles. And that would be from the capsid protein sequences from the prototype or typical virus strain that we use in the laboratory.
Thank you, Dr. Berger. There was another raise of hands straight on at the very end. Is that still valid, or no? No longer. Here in the middle of the.
Okay,
Yeah.
I'm Albert van der Zijden. I'm co-founder of the Dutch Crohn's Ulcerative Colitis Association and of the European Federation of Crohn's Ulcerative Colitis Associations. And I'm also a patient with ankylosing spondylitis, so I have an interest in the issue. But what puzzled me is the fact that if you look to the distribution of reported cases, they are almost all in the United States or North America and the European Union. Is there a reason for that? Are the medicines not used in other parts of the world, for instance?
To whom was the question directed?
To all speakers.
To all speakers. Oh, that is a challenge.
Maybe one of them knows it.
Brigitte.
Yeah, Brigitte.
There are, with regard to Tysabri, there are 5 cases reported from Switzerland. If you consider the population, the size of the population, so that is comparable to the reporting rate from other European countries. And there was 1 case from Australia. Yes, there is reporting from other countries. And as I remember also for the other monoclonal products. But it's often the case that in the European Union and in the US, the reporting rate is higher than in other areas of the world. So yes, but there is reporting also from other areas.
Any other comments from the speaker side? No? Okay. Declan, you would like to ask something?
Thank you, Christoph. And it's an initial question for Dr. Brigitte, please. I'm representing the Deferno Trust, which I set up as my wife Natalie is one of these statistics. She is the 36th case of Tysabri-related PML to be diagnosed. And I suppose if we look at the scale we talked about earlier on, she's probably in the 20 to 30% range, where she is in a full paraplegic state and does not have the ability to communicate either. I just want to thank Christoph for actually being involved here this morning. The question I specifically have is 2 parts. One of the major issues that we have as a family and as a patient group is the information availability. And it was very, very refreshing to hear this morning just this information that has come to light. 2 questions I have: who actually, on what database was this information derived from? And the second part of it is there any plans to have this information being made available in a real time in the public domain for us as medical professionals, their patient groups and their families?
I think that's a question to you, Brigitte. Or no?
Probably you're better off.
I think that there's an intention of EMA also to publish results of this workshop. That is the first part of the answer, and I see nodding heads. And second is that CHMP requested from the marketing authorization holder to present some of the data on the web page. But of course, and maybe that Peter can help me from the side of EMA, that we will publish these data. These data are derived from the user vigilance database and from the Paul-Ehrlich Institute's database. And I have to
mention also that the marketing authorization holder in the European Union is requested to frequently submit to the CHMP and to the rapporteurs safety data and analysis of this data. So yes, this data is available in the EUDR Vigilance database, and I think, Peter, this data will also be published.
If I may, Christoph.
Sure.
So Biogen has been kind enough to provide on a monthly basis the updates to healthcare providers that have requested it. So as a healthcare provider, I've been made a certain amount of information anyhow on a monthly basis. And I know that's true elsewhere. I know in Germany, and I suspect in other countries, the healthcare providers can get that information from the company. Now, it doesn't tell you everything, and it is an enormous source of frustration that we don't have real-time data, as much real-time data as we'd like. But then again, it's very difficult to assemble that information. As for having a website, you're to be congratulated to have a website available to educate people about PML. I will tell you that somebody uploaded a talk on PML I once gave years ago on, I think it's on YouTube. And probably every 3 months, a patient calls from somewhere and says, "Can I come see you?" Kentucky's not the easiest place to get to, by the way. And I say, "Of course." And the only way they knew about me was because they'd seen me on YouTube. Rather curious.
Just if I may follow up, Christoph, if you don't mind. The issue that we have, as we see it, the information is coming, let's say, to one stage, but the information that I get is being trawled through the financial and medical press. And as a patient, a representative of a patient, it is very, very frustrating that we get this information late on a Friday evening or a Monday morning or whatever the case may be. And actually the information that I have currently where I have is coming from another husband of another PML patient who is, dragging this information out. Now, it may be made available, as I said, via the pharmaceutical companies to the healthcare professionals. It's not coming down to the families who really, at this stage, are looking for every piece of information they can get to try and help them understand this at such a traumatic time. So, again, it's just a step forward. The information today is absolutely valuable. For me, at least I can see something that's happening. But as I said, hopefully something more positive can come out of today.
Thanks, Declan. We will probably address the issue of information to patients and carers at the end in the session 8 when we come to the questions, what is coming out next. But do you want to give a short comment, Peter?
Well, just to say that one of our objectives from this workshop is to look at better ways of sharing information post-workshop. We will focus on that question, particularly in session 8. And I think what we would like to achieve is not that we set up something that is just a one-off that then isn't maintained, but that we look at ways of leveraging things that are already out there, the website and the blog that was already highlighted earlier, for example, and how can we sort of fill gaps in terms of information exchange going forward in a sustainable way. So we'll come to that tomorrow afternoon.
Okay, thank you. Well, looking at the watch, we are running late a little bit, but we still have enough time for a short summary by my co-moderator, Dr. June Rain.
Thank you very much, Christoph. Just a few wrap-up comments. I'm sure you will agree that this overview of the current PML scenario has brought together a fantastic range of up-to-date information on disease mechanisms, on outcomes, on contrasting regulatory perspectives, the drugs and the risks in different patient populations. And I know it's our tendency always to focus on what we don't know, but let's in these 2 days make the most of what we do know and what we can bring together. I think I can perhaps best summarize Jean Major's comments as by saying, pay attention to the virus. Let's all pay attention to this virus, the absence of animal models, the archetype not growing in the lab, the fact that no 2 PML patients have the same sequences for the viral capsid protein. Let's pay attention. On outcomes, we've been thinking hard about this variability we're hearing about in terms of survival. What can we learn from the different epochs of PML? But there are clear common themes which Joe Berger has set before us to think about and to build on. We've heard some fascinating information about the contrast between the EU and the US, and we must think hard about this, what that is telling us from Brigitte, particularly in relation to Tisabri, the calls for international standards, for agreed criteria for long-term follow-up, for better stratification of risk. Surely we can begin together to tackle these. And then we were broadened by Professor Dupasquier to other drug-associated PML, the use of the WHO database to help us, but what about potential underreporting and notoriety bias? How can we address these? And finally, let's think about the transplant patients and what we can learn from this patient population with the potential link to transplant rejection. But maybe we should finish, as we have done in our discussion, on the patient perspective. As Christoph has said, on the value of partnerships. We are here to work in partnership. If there is up-to-date, useful information, let's make sure that it flows to all those who need to benefit from it in real time. We heard good news about the support in Europe for an MS register, but maybe this is just one step. There needs to be so much more in relation to the potential for internet-based networking. So I'll close my remarks by thanking our excellent speaker panel for doing all this work on our behalf and setting us for 2 days of really productive and constructive debate. Please join me in thanking them.